Hi everyone. Today we're going to be solving AQA GCSE Biology Higher Tier Paper 2H, June 2021. The nucleus of a cell contains DNA. Name the structure inside the cell nucleus that contains DNA. So if we look into a typical cell, this is the nucleus, and inside the nucleus we're supposed to have chromosomes. So DNA is contained within the chromosomes. So we'll say chromosome. Now this is where we're going to find the DNA. Figure 1 shows part of a DNA molecule. We can see figure 1. We are shown the phosphate and adenosine, thymine, cytosine, and guanine. So this is adenine, thymine, cytosine, and guanine. Now guys, you need to remember that A always pairs with T and C always pairs with G. These dashes actually represents hydrogen bond between them. All right, this is the sugar. So because this is a DNA molecule, so this will be deoxyribose sugar. Name the part of the DNA molecule labeled X. So X will be the sugar. What type of substance is labeled ACGT or ACGT in figure 1? So type of substances. ACGT, these are bases. So we will just say bases. They are mainly nitrogenous bases. So I'll just keep the name here so that you can learn from it. Figure 2 shows another section of DNA molecule. We can see this DNA molecule is unwinding, unzipping or unwinding. Then we can see new nucleotides are joining here. So the whole process here that is taking place is called DNA replication. All right, now let's see. 4 of the substances you named in question number 1.3 are not labeled in part Y in figure 2. Label each of the substances with the correct letter ACGT. Use information from other parts of figure 2 to help you. So, as we discussed already, C pairs with G and A pairs with T. So, wherever A is written, we're going to write T. G opposite will be C. C opposite will be G. And T opposite will be A. What is happening to the DNA molecule in part Z in figure 2? As we already talked about earlier, replication is taking place. DNA replication is the process of unzipping and then adding new nucleotides to make 2 strands from a single strand. So it is the process of making 2 DNA molecules from 1 DNA molecule. Fertilization is the fusion of gametes. So egg cell and sperm cell fusion is fertilization. Evolution is the process of forming a new organism from an old organism by means of natural selection. Differentiation is the process of specialization. So specialized for a particular function. A gene is a length of DNA. What type of substance does a gene code for? So gene codes for a polypeptide or a protein. Most human body cells contain 6 times 10 to the power of minus 12 grams of DNA. What mass of DNA will a human sperm cell contain? Since human sperm cell contains half number of chromosomes, which is 23, and normal body cells contain 46 chromosomes, so it will be half of this value. So 3 times 10 to the power of minus 12 grams. What is the name of the type of cell division that produces sperm cells? Binary fission is for bacteria. Is the bacteria's way of cell division. We already talked about differentiation. It's a form of specialization. Meiosis is during sexual reproduction. It produces gametes such as sperm and egg. So this will be the correct answer. Mitosis produces genetically identical daughter cells. A student investigated the effect of temperature on the decay of milk. Figure 3 shows the apparatus the student used. We can see figure 3. It consists of a pH meter, fresh milk with a pH sensor dipped in it, and a thermometer to measure the temperature of the milk, and a computer which is used as a data logger. This experiment is looking at the decay of milk. In biological term, decay means to break down something into simpler substances. This is the method used. Set up the apparatus as shown in figure 3 with the milk at 20 degrees Celsius. Record the pH over 5 days using the computer. Reboot with another batch of fresh milk at 25 degrees Celsius. How could the student keep the milk at constant temperature for 5 days? To keep the milk at constant temperature, a water bath could have been used. So we can put the beaker in a water bath and then maintain the temperature of the water bath. Give one variable the student should keep constant. Do not refer to temperature in your answer. Now the reason why they say do not refer to temperature, because this experiment is looking into changes in temperature and rate of decay. So in every experiment, of course, the volume of milk should be the same and the type of milk should also be the same. So we will say either volume of milk or type of milk. Table 1 shows the student result for the milk at 20 degrees Celsius. We can see time in days from 0 to for 5 days it was measured and the pH decreased from 6.7 to 4.4. Complete figure 4. Okay, we have to label the x-axis, use a suitable scale for the x-axis, plot the data from table 1 and draw a line of best fit. So our x-axis will be time in days, and since there is a total of 5 days, so we can start from 0. At 0, the value reads 6.7. At 1, the value still reads 6.7. At 2, the value reads 6.3. At 3, the value reads 5.3. At 4, 4.6. And at 5, 5.4.4. We also have to use a line of best fit. So let's join the lines together. The data you plotted in question 2.3 were obtained at 20 degrees Celsius. Sketch a line on figure 4 to show the result you would expect at 25 degrees Celsius. Label this line 25 degrees Celsius. So at 25 degrees Celsius, the reactions are going to be faster. So it decays faster. As long as our line is below the original line and comes up and levels up with this particular line, we will be more or less correct. Human body temperature is controlled within narrow limits. Scientists investigated the effect of drinking ice-cold water on internal body temperature, the rate of sweating. This is the method used. Sit a person inside a room kept at constant temperature of 25 degrees Celsius. Measure the person's internal body temperature near the brain. Measure the person's rate of sweating. After 20 minutes, give the person 500 cm³ of ice-cold water to drink. Continue to measure the person's internal body temperature and sweating rate up further 50 minutes. Our body temperature is controlled within very narrow limits. Our internal temperature, it can go as low as 36.5 and as high as 37.5 on a normal day. So usually 36.5 will be on a cold morning and 37.5 will be on a hot day. Give the reason why the person should not move during the investigation. Because movement causes muscle contraction, so there will be extra heat that would be released. So we can write movement would release extra heat, or movement would increase body temperature, or movement would increase sweating. Figure 5 and Figure 6 shows the scientist's result. The person drinks the ice-cold water. His temperature goes from 37.4, it goes as low as 36.8, and it takes about 14 minutes. And then the temperature rises to 37.4 back again. And that happens at 60 minutes. So that takes about 26 minutes. Rate of sweating in arbitrary units. The person drinks at 20 minutes. Rate of sweating decreases, goes down to 0.9, approximately 36 minutes. So 16 minutes after drinking the ice cold water, and then rises to the same level back at 56 minutes. So 20 minutes. All right. The sweating rate again increases back to original 3.7. What is the person's normal internal body temperature? The normal internal body temperature was 37.4. The results show that when ice-cold water was drunk, the temperature near the brain decreased. Explain why the temperature near the brain decreases. So, when the ice-cold water is drunk, two things happen. It goes into stomach. Now, the stomach temperature decreases, which in turn decreases blood temperature in the stomach. So, this blood then returns to the heart and then gets pumped to the brain maybe. So we can write down the answer in this way. The blood is cooled in the stomach or the mouth, and the cooled blood flows to the brain. The thermoregulatory center in the brain responds to the decrease in temperature. How does the thermoregulatory center sends information to the sweat glands in the skin? So thermoregulatory center is connected to the sweat glands via nerves, and nerves communicate by impulses. So we'll write down the answer via nerve impulses. The rate of sweating changes between 24 minutes and 36 minutes. Explain how this change helps to maintain the person's normal body temperature. So we can see at 24 minutes, it starts going down. Sweating rate starts decreasing. So sweating decreases, and sweating by itself helps to decrease body temperature. So we can write down the answer. Sweating decreases from 24 minutes to 36 minutes. So less sweating occurs, and as a result, less heat is lost by the evaporation of the sweat. During exercise, the skin appears red. What causes the skin to appear red? So during exercise, vasodilation occurs in the skin. This helps to reduce blood temperature by radiation. So let's see the options. Blood vessels moving closer to the skin surface, constriction of blood vessels in the skin, decrease in heart rate, dilation of blood vessel in the skin. So obviously dilation of blood vessels in the skin. So why does the dilation take place? To increase heat loss by radiation conduction. Decay occurs in a compost heap. Figure 7 shows a compost heap. We can see in this compost heap there are thin layers of soil, and in between those layers there are layers of dead plant materials. Describe how microorganisms in the layers of soil help to recycle chemicals in the dead plants. So when we are referring to microorganisms, we are mainly thinking of bacteria and fungi because they are mainly decomposers. How the chemicals are used by living plants. So then the dead plant material will decay to release CO2 and minerals. Our next portion will be to use a living plant, how those CO2 and minerals are recycled into new plants. This question will require a level 2 answer. So a level 2 answer needs to be scientifically relevant fact. We have to give details as well. So the answer can be written as follows. In microorganism, digestion breaks down large molecules into smaller molecules by the help of enzymes. Then respiration of small molecules releases CO2. Breakdown of nutrients liberates the mineral ions into soil. Example, nitrate, magnesium, phosphate. In plants, carbon dioxide from the air is taken up by the leaves by diffusion via the stomata. CO2 is used for photosynthesis to make sugar and cellulose. Mineral ions are taken in by roots by active transport. Nitrate is used for making proteins. Phosphate is used for making DNA. The growth of daisy plants on a lawn is affected by biotic factors and the abiotic factors. Table 2 shows 6 factors. Tick one box in each row to show whether the factor is biotic or abiotic. When talking about biotic factors, these are the living factors within an ecosystem. And abiotic factors are non-living factors. So nitrates in the soil is a non-living factor, so it will be abiotic. Rabbits eating the plants. Rabbits are living organisms, so it will be biotic factor. Shading by a building. Shade is not a living factor, so abiotic factor. Soil pH. Soil pH is the acidity of the soil, and that is not a living factor, so abiotic. Temperature is also abiotic because it's not a living factor. Trampling by people. Since people are living organism, so biotic factor. Figure 8 shows a plan of a garden. We can see there is a paved area, but other than that, the garden is a triangular garden with 10 meter by 16 meters. A student estimates the number of daisy plants growing on the lawn. The student places a quadrant at 10 different positions on the lawn. The quadrant measures 50 centimeter by 50 centimeter. A quadrant that measures 50 centimeter is 0.5 meters. Since it measures 0.5 meters by 0.5 meters, the quadrant is 0.25 meter squared. The student then count the number of daisy plant in each quadrant. How should the student decide where the quadrant should be placed? Give the reason for your answer. The student needs to place the quadrant randomly. To do it randomly, the student can make the entire field into grid and then find coordinates, all right, using random number generating app. This will achieve randomness. The mean number of daisy plants in each quadrant is 6. Calculate the number of daisy plant on the lawn. Give your answer to 3 significant figures. First of all, each quadrant was 0.25 meter square. So we will require how many plants in 1 meter square. So since there are each quadrant is 0.25 meter square, so in 1 meter square, there is 4 quadrants. And then 4 quadrants multiplied by 6 gives us 24 plants. Now we need to calculate the area of the lawn. We can use the formula half times length into breadth. However, there is a small complication because the lawn contains a paved area. So we will have to subtract for the paved area. So the area comes 80 minus 18, 62 meter square. Since there are 24 plants in 1 meter square, so number of daisy plant will be 24 times 62, 1488. Our answer needs to be up to 3 significant figure. So 1488 will be written as 1490. Using the mean from this investigation to calculate the number of daisy plants on the lawn may not be accurate. We have done the mean from 10 quadrants. This is very limited in number. And each of the quadrants, and those quadrants were only 0.25 meter square. So might not be representative of the whole area. So the question asks, give 2 reasons why. So one of the reasons we can say too few quadrants used, or the quadrants are too small. And the second point we can write, the sample may not be representative of the lawn, or the quadrants may not have been placed randomly. Reflex actions are coordinated by the nervous system. What is meant by the term reflex action? A response or a reaction which is automatic or does not require thinking. A woman's hand accidentally touches a hot object. The woman moves her hand away rapidly. Describe how the woman's nervous system coordinates the reflex action. So, the hot object or touching the hot object will be detected by the receptors in her hand, and then the receptor will generate an impulse which will travel down through her sensory neuron to relay neuron, which is in the spinal cord. And then from the relay neuron, it will be conducted to the motor neuron. From the motor neuron, it will be conducted to the muscle. And the action will be removing hand. Now, in the sensory relay and the motor neuron, these will be impulses because they are neurons. However, the bridge between the sensory and the relay neuron will be synapses. And this bridge will be crossed by using chemicals such as neurotransmitters. Chemicals. So the answer can be written in this way. The receptors in the skin of the hand detects the stimulus, or we can say the temperature change. The impulses then travel along the sensory neuron to the relay neuron to the motor neuron. And in between the neurons, there are synapses where chemical crosses the gap, as you can see in the picture. The synapses are located in spinal cord. The motor neuron brings the impulse to the muscle, and the muscle contracts, which pulls the hand away from the hot object. The endocrine system coordinates many internal functions of the body. If 3 ways coordination of the endocrine system is different from coordination by the nervous system. Coordination by endocrine systems are hormones, which are released directly into blood. And nervous system sends impulses along neurons. Since hormones are directly released into blood, this system often is much slower in comparison to impulses, which is faster. However, hormones are released into the blood, so it stays in the blood for longer, so we'll say long-lasting, in relative comparison to impulses, which is for a few seconds, so short-lasting or a single response. So the points we can write, hormone is slower, but it is longer-lasting, and hormone is released via blood. Describe how hormones control the menstrual cycle. When thinking about the menstrual cycle, the few hormones that we should think about are FSH, followed by that LH, in between them estrogen, and after LH progesterone. So let's see each of their function. FSH, follicle stimulating hormone. So it stimulates the maturation of an egg cell or follicle. Estrogen repairs endometrium or uterus lining. Progesterone, the function is to maintain uterus lining. LH is responsible for ovulation. So we can write down the answer in this way. FSH released from the pituitary gland stimulates the maturation of egg, whereas estrogen released from the ovary inhibits the FSH production. Now, this is inhibited because we do not want multiple eggs to be produced, and stimulates the LH production. LH is released from pituitary gland, which stimulates ovulation, which is pretty self-explanatory because estrogen does not want multiple eggs to be matured and released, and LH is responsible for ovulation of the egg. Progesterone released from the corpus luteum inhibits FSH and LH. Progesterone is also needed after pregnancy. So, progesterone inhibits both FSH and LH so that multiple eggs are never released. And both estrogen and progesterone maintains the uterus lining. A food web contains several food chains. Figure 9 shows a food web. So a food web shows feeding relationship within an ecosystem and their interdependency. In this food web, the algae is the producer. This makes the krill a primary consumer, copepod a primary consumer, herring also a primary consumer, but secondary consumer with respect to copepod, and so on and so forth. Where we can see human is one of the tertiary consumer or apex predator. Another apex predator represented is cormorant. An apex predator is a top predator that doesn't have any organism fitting on it. The animals in figure 9 get their energy by eating other organisms. Describe how the algae get energy. So algae are photosynthetic organism. So they receive their energy from sunlight and they absorb the sunlight by using their chlorophyll. Name one primary consumer in figure 9. So primary consumer in figure 9, as we already discussed, are krill and copepod because they both feed on algae, and also herring because herring feeds on algae. So any one of the name will be correct. We just need to write one of them in the exam to get the mark. Name one producer in figure 9. So the producer in figure 9 is algae only. A producer is an organism that does photosynthesis to produce organic molecules like sugar or amino acids. The different food chains in figure 9 have different number of organisms. Complete figure 10 to show a food chain in figure 9 with 5 organisms including human. Since the key point here is 5, and also we have to include human as the apex predator. So considering that algae is the producer, which is first part, then we can move to krill, which is the second, which is the primary consumer. Then we can move to squid, which is the secondary consumer. Then we can move to mackerel, which is the tertiary consumer, and then human, which is the quaternary consumer or apex predator in this case. So total 5 steps. Let's see again. Algae is the producer, krill is eaten by squid is eaten by mackerel, and then human. Figure 9 shows that mackerel eats krill and squid. The biomass of mackerel is much less than the combined biomass of krill and squid. One reason for this is that mackerel cannot digest all parts of the krill and squid. Give 2 other reasons. So already inedible parts are mentioned or undigested portions are mentioned. So other points we can say is respiration, because energy is lost as a respiration. Some of the parts are not eaten from the squid and the krill, or we can say energy is lost in excretion by the mackerel. Figure 11 shows how the biomass of adult herring in the North Sea has changed between 1950s and 2010. We can see the biomass of adult herring in millions of tons. Overall, there is a decrease from 1950s up until 1977. And then there is an increase from 1977, up until 2010, general increase. Calculate the percentage decrease in biomass of herring between 1960s and 1997. We have to give our answer to the nearest whole number. So at 1960s, the biomass was 2.3 million tons. And in 1977, the biomass was 0.1 million tons. So 2.3 minus 0.1 divided by 2.3 times 100. This gives us 95.65, which is equivalent to 96. To score the full mark, we'll have to represent the 2.3 million and 0.1 million in the box here. So 1960s, 2.3 million tons. 1977, 0.1 million tons. Too many herring were caught by fishermen between 1960s and 1977. Herring can live for up to 12 years and begin to reproduce when 3 to 4 years old. Laws have been introduced to help conserve herring. So here the conservation means to protect them so they're able to reproduce. 1917 to 1981, herring fishing was banned in the North Sea. 1984 to present day, control of mesh size of fishing nets. So the control of mesh size of the fishing nets allows smaller fish to escape so that they can reach their adulthood and breed. Banning fishing stops the fish from getting, decreasing in number overall by humans. So 1997 to present day, fishing quotas were introduced. Fishing quotas is a method of limiting the amount of fish that can be taken. So this in turn allows brood stock to remain in high number. 1998 to present day, herring fishing was banned in breeding grounds during the breeding season. So this protects the egg-carrying female. Figure 12 shows how a minimum mesh size helps to conserve herring. So the 8-year-olds are getting caught, which are matured enough, and the 4-year-olds are not getting caught since they are small enough to pass through the fishing net. So they will be able to breed up until they are 8 years or so. Figure 11 is repeated below again, and the question asks, evaluate the effect of these laws on the conservation of herring stock. Use data from figure 11 and information from figure 12 in your answer. So 1977 to 1981, when fishing was banned totally, we can see the amount of fish increased dramatically. In 1984, the control of mesh size, the fish kept on increasing. In 1997 to present day, fishing quotas were introduced and we can see the fish decreased in numbers, however then recouped back to higher numbers. So the answer can be written as follows. Small fish are not caught so they can live long enough to reproduce. The biomass have gradually increased after these laws. From 1977 to 1981, total ban resulted in increase in biomass from 0.1 to 0.48. 1984 law mesh size resulted increase in biomass from 0.9 to 1.8 by 1990. The number could have increased due to other causes. Each law results in increase followed by decrease. Quotas lead to dead fish being thrown back at sea. Sickle cell anemia is an inherited condition that affects the red blood cells. Sickle cell anemia is caused by mutation in the gene for hemoglobin. Hemoglobin is the red pigment found in red blood cells. A person who is homozygous for the normal hemoglobin, HA, produces normal red blood cell. A person who is homozygous for the mutated HS produces red blood cells with abnormal hemoglobin, has red blood cells that can form a altered shape, has sickle cell anemia and becomes ill. A person who is heterozygous has both a normal and abnormal hemoglobin in the red blood cell, has sickle cell trait, is generally healthy but can become ill in certain circumstances. So a person who is heterozygous has HA and HS both. A person who is homozygous with the mutated allele has HS and HS. So a normal person would be homozygous with HA and HA. Give the reason why mutation in the gene coding for hemoglobin could be harmful. This mutation means the person can carry less oxygen for aerobic respiration. Figure 13 shows some red blood cells from the blood of a person with sickle cell trait. So again, sickle cell trait means the person has HA and HS, means the person is heterozygous. As you can see from this picture, some red blood cells are sickle shaped, whereas the rest of the red blood cells are normal. Calculate the proportion of the cells in figure 13 that have an altered shape. So we can see 1, 2, 3, 4. 4 has altered shape out of 17 cells. So 4 divided by 17, 0.235, which is equivalent to 0.24 or 24%. Figure 14 shows the inheritance of sickle cell anemia in one family. So if a male has sickle cell trait and a female has sickle cell trait, means they are both carrier of sickle cell. And if they have sickle cell anemia, that means they have homozygous sickle cell allele, and unaffected male and female has both normal. So homozygous for the normal allele. Person 8 and 9 in figure 14 are expecting a second child. So let's look at person 8 and 9. Person 8 does not have sickle cell anemia and he is a male. So HA, HA, whereas person 9 is carrier, so HAHS. They will have another child. Determine the probability that the child will be a girl and with the sickle cell trait. Draw a Punnett square diagram. Identify the phenotype of each offspring. Genotype. Use the symbols HA for normal hemoglobin and HS for mutated hemoglobin. So father is 8, which is HA as HA in both of his alleles. Mother is a carrier, which is 9, has HA plus HS. So let's do the Punnett square now. Sorry, father has HA and mother is HAHS. This gives us, so we can see 2 children out of the 4 has the chance of having sickle cell trait, 2 over 4 or 0.5. Now, having a offspring which is female, probability of a girl. Father is X and Y and mother is X and X. So the probability of having a girl is also 2 over 4, means 0.5. Probability of having a girl with sickle cell trait will be 0.5 times 0.5 equals to 0.25 or 25%. Without medical treatment, people with sickle cell anemia are frequently ill and have a reduced life expectancy. The malarial parasite cannot live in red blood cells of a person who has HS allele. The scientist stated it is an advantage for people to have HS allele in countries where malaria occurs. Evaluate the scientist's statement. So, the way we are going to write this particular answer is that a person who is heterozygous HAHS does not get malaria and can survive sickle cell anemia. And then we will write down H. If the person is HAHA, then the person may die from malaria because the person can be infected by malaria. But if the person is HSHS, then the person may become severely ill with sickle cell anemia. But even though the person does not get malaria, the person will not survive sickle cell anemia likely. So if parents are HAHS, then some offspring will survive both malaria and sickle cell anemia. We will need only 3 points to score this mark. The Galapagos Islands are located in the Pacific Ocean. Several species of bird called finches live on the Galapagos Island. These finches are very similar to each other. Figure 15 shows the modern species of Galapagos finch and their classification. We can see medium ground finch and small ground finch. So their classification group is mentioned and we can see there are few missing ones. So to fill up the missing ones would be our mark given in the first question. It is complete figure 15. Give the names of the missing classification group. To do the missing classification group, we can remember the mnemonic. King Philip comes over for good spaghetti. So the king refers to kingdom and Philip refers to phylum. And comes refers to class and over refers to order. F refers to family. Good refers to genus and spaghetti refers to species. If you remember the mnemonics during your exam, you will not get this wrong. Give the binomial name for the medium ground finch. Use information from figure 15. So we need the binomial name. It refers to a two-part name. The word bi means two. And the first part will be the name of the genus. And the second part will be the name of the species. So the name will be Geospiza fortis. In each species of finch, there is a variation in beak depth. Figure 16 shows how beak depth is measured. Figure 17 shows the relationship between beak depth of parent birds and the beak depth of their offspring. Mean beak depth of parents in millimeter and mean beak depth of offspring in millimeter. As you can see, if one of them is increasing, the other one is increasing. So it is a positive correlation. Give evidence from figure 17 that the beak depth is a inherited characteristics. So the answer will be offspring have similar beak depths to parents. That means it's inherited. Scientists suggested that more than one gene controls beak depth. Give evidence to figure 17 to support the scientist's suggestion. So we can deduce from the graph that parents of a given beak depth can produce offspring with several beak depths. Figure 18 is a map of Galapagos Islands. So we can see multiple islands in the diagram. On Isabella Island, the medium ground finch and the small ground finch are found. On Daphne Island, only the medium ground finch is found. On Crossman Island, only the small ground finch is found. Figure 19 shows how the beak depth of each species varies on each island. So we can see beak depth in millimeter and percentage of population. Small ground finch has a smaller beak depth, all right, and 8 being the highest in percentage, and medium ground finch has a broad range of beak depth and 12 being the model class. Percentage of population, beak depth in millimeter on Daphne Island, 10 is the model class, and it ranges from 8.5 to 11.5 or a little bit higher. Depth in millimeter for Crossman Island ranges from 8.5 to 10.5, and percentage of population, all right, we can see highest one is with 10 or 8. The second highest is 8. The medium ground finch and the small ground finch both feed on seeds. The size of seeds eaten by each bird depends on the depth of the bird's beak. The range of beak depth of medium ground finches on Isabella Island is different from range on Daphne Island. Explain what might have caused this difference. So the answer can be given as follows. The colonizers of Isabella have a range of beak depths due to different combinations of alleles of several genes. Large range of size on Isabella due to size of the seed available in Isabella. There is more competition for seeds on Isabella, so birds with larger beak get enough food to survive and reproduce, and the survivor then passes on the beneficial alleles to the next generation. Figure 19 shows two species of finch live on Isabella. Only one species lives on Daphne Island and only one species lives on Crossman Island. Suggest why both species of finch are able to live on Isabella Island. So, looking back into the information of Isabella, we will see that they do not have a, crossing beak size, meaning in Isabella Island, the medium ground finch has different food compared to the small ground finch. So not same niche or the same food. Now, when the question says suggest why both species of finch are able to live there, one of the answer that could be given is that Isabella Island, from the diagram also we can see it's very large. So we can say Isabella Island is large island with more species of plants. So there is enough seeds for both of the bird species. So guys, that's the end of this particular question paper. Thank you for watching the video, guys, and best of luck for your exam. Subscribe to the channel, and question paper, videos like this will be always uploaded in the channel almost every day. So guys, subscribe to the channel and let the channel grow. Hope this question paper solving helps your exam. Thank you guys. See you in the next paper. Bye-bye.